I want to talk a little bit about potency, persistence, and mechanism of action, or MOA. We've talked through what makes your new NK platforms stand out. I want to dig into some of the tougher questions, like how we tackle the big issues of persistence and how we stay safe for patients. And so, Raphael, for you, we know that persistence has long been a concern for NK cell therapy, but how does your technology overcome the challenge and convert NK cells into these effective serial killers?
Yeah, so I think at the end of the day, everything is relative in some way. What matters is the balance between the ability to kill enough cancer cells to eradicate the tumor and to long last enough so we can do that in a continuous way, knowing that the safety outcome is also critical on the treatment that you give. So it's always about a balance. So we explained already, I did, and Tracy with different approach, but we both have a main goal is that the paradigm and the dogma that says NK cells die out after 2 weeks, we are beyond that. This is it. We have found ways to prolong the life of NK cells, and I'm going to insist is to functionally prolong the life of NK cells. I don't care to have NK cells present in the blood 6 weeks after. I care about having those NK cells actively killing 6 weeks after they have been infused. So it is really important, and this is why we have a responsibility and our science team to provide the data that is showing that it's not just, hey, look, you can find my NK cells in a patient 6 weeks after. No, it's like you can see it and they are still killing. That's really important because it's like I'm sorry, but I don't care to see NK cells present. I care about seeing them actively killing. Now, the potency aspect, I think there are so many different approaches today that are actually all of them bringing very interesting potential outcome. Like I said, there are multiple labs, multiple biotechs working on NK cell. Unfortunately, less and less of them because many either have abandoned or pivoted to other type of technologies or just unfortunately bankrupted because this is the reality of our current business. A lot of companies are filing bankruptcy because they cannot fundraise. But what is really important is when you develop a new technology, you need to validate it. You have a set of different experiments that needs to be done, and you need to tick those boxes. You need to show first it has to kill, then it has to reach the tumor, because unfortunately sometimes you have fantastic data in vitro, and then you go in vivo and your cells are just not getting to the tumor. They're not going there. And if they don't go there, how are they going to be able to kill? So there's a second aspect, which is known to be the trafficking question. And some companies have failed, unfortunately, because their trafficking was not good enough. Then once they are in the tumor, and you think about solid tumor in particularly, how do they penetrate that tumor? How are they really able to go deep dive into the tumor so they could kill more tumor cells, or are they're just going to kill what is presented to them in the beginning, and how they're going to be able to go over this? Then in the solid tumor comes the question of tumor microenvironment. The tumor microenvironment, and this is high in the poll also, is known to be potently immunosuppressive, which means it shuts down the immune system to protect the tumor. And interestingly, our technology brings something very unique because one of the key contributor of immunosuppression is HLA-G. And HLA-G is heavily present around the tumor and in the tumor microenvironment. And our approach is that not only are we going to be able to use it to target the cancer cells, but if we can shut down HLA-G, we can reduce this immunosuppressive effect that it has, which can allow us to eventually have more opportunities to recruit other immune cells, whether you use a combination treatment or you just have the immune cells no longer being shut down by the tumor microenvironment and able to attack the cancer cells. So it's never just a one point and that's it. There's so many parameters that are coming that makes it extremely complicated. But I think the route of enhancing the potency to make our cells stronger killers and make them really, I insist on the terms of serial killer, able to, once activated, not only kill 1 or 2 tumors when they cross paths, but chasing, going to the tumor site and killing, and doing this for a duration that is more than 2 weeks, 3, 4, 5, 6 weeks, maybe more. This is the right combination of events that need to happen to have an impact on cancer cells. Everything else will have to prove its case once we get to patients. We will prove it in vivo. We will show that it works in mouse models. We actually just finalized a partnership with a major institution to work on PDX models with them and to prove that our technology works across multiple different PDX model, which is the use of real tumors that we put into mice to be able to check how our cells are working on it. And all this evidence are needed to reassure the regulator and the regulatory bodies that we can go to patients. But then the answer will only come once we treat patients. And Tracy knows that better than anybody because they do treat patients already. This is where we're going to see if our technology works. This is where we're going to see if all the hypotheses that we have validated can be replicated in a human being. And if they can, then we have a shot. Then we have a shot at getting enough efficacy, proper safety to get the drug approved and to then expand it to multiple patients globally. So it's a tedious and long process. The people on the audience, I know they all know about this because they are likely all experts in the field of biotech. But we have to answer hundreds of questions at a time, and we have to take them one question at a time to be sure that we answer it properly.